This video is part 1 of 4 for Chapter 3, the cellular level of organization. As we mentioned previously, one of the functions of living organisms is maintaining boundaries. Just like we have external boundaries, our skin is a great example of an external boundary, we also have internal boundaries. At the cellular level, the boundary for each cell is known as the plasma membrane. It helps to separate intracellular fluid from extracellular fluid. Intracellular fluid is the fluid internally inside the cell, and extracellular fluid would be that surrounding the cell. The plasma membrane is referred to as a semi-permeable membrane, and that means that some things can pass through and other things cannot. This controls the entry and exit of different things like ions, nutrients, waste, and secretions. Recognition and response to molecules internally and externally can be regulated by the plasma membrane. The plasma membrane also lends structural support and connects cells to other cells, giving our tissues stability. All cell membranes or plasma membranes maintain what's known as a resting potential, which is excitable for cells. This resting potential is actually an electrical difference that can be measured, which you can imagine would be really important for neurons to transmit signals, as well as for muscle cells in order to be able to contract. Again, we'll see that very soon. One of the major components of plasma membranes or cell membranes is a molecule called a phospholipid. Phospho refers to the phosphorus group or phosphate group of this molecule, and that makes that portion of the molecule polar. Because it has polarity, it likes water, therefore is known as being hydrophilic, water-loving. The two fatty acid tails of a phospholipid are referred to as hydrophobic because fat and water do not mix. Phospholipids make up the plasma membrane as they are arranged in something that is known as a phospholipid bilayer. That means there's two layers of phospholipids. On the lower left picture, we see that these phospholipids are arranged in such a way that the hydrophilic, water-loving polar phosphate heads are facing the extracellular fluid externally, as well as the intracellular fluid internally, whereas the hydrophobic fatty acid nonpolar tails are arranged on the internal portion of this plasma membrane. That means that things that are polar, like ions are polar because they have a charge, and water-soluble components can't cross that membrane very easily. They might leak through, but for the most part, they don't cross the membrane easily, whereas fat-soluble molecules, including oxygen and carbon dioxide, can easily pass through the membrane because they are fat-soluble. So anything that is fat-soluble, like oxygen and carbon dioxide, can cross through easily. That makes a lot of sense, and it's very helpful for oxygen to come into the cells without needing any kind of special carrier, and for carbon dioxide to exit the cells. Looking more closely at this phospholipid bilayer, at the picture on the upper right-hand side of this slide, we notice that there are some cholesterol molecules embedded in this phospholipid bilayer, and that helps to stabilize the bilayer. Remember, these are molecules that are attracted to one another, so they're moving around slightly. It's what's referred to as a fluid mosaic model. So things are moving around slightly. They're attracted to each other, and that holds them together. The attractions hold them together. However, there is some movement, and cholesterol molecules help to stabilize the membrane. We also see embedded in this membrane an integral protein, which will span the length of the membrane, a peripheral protein. Peripheral proteins may be found on either side of the plasma membrane. We also notice a glycoprotein. Glyco meaning sugar, protein is a protein. So a glycoprotein is just a protein with a sugar attached to it, and here they actually point out the carbohydrate. We also have a glycolipid, which is a fat molecule with a carbohydrate attached to it. And we also see fibers of the extracellular matrix. The matrix, that particular term refers to the stuff surrounding a component, whether it's a cell or anything else. The matrix is just the stuff surrounding a certain object. Looking more closely at membrane proteins, these proteins allow communication between the cell and its external environment. Membrane proteins account for about half of the mass of a plasma membrane, and they have very specific functions. Some are going to kind of shift through the membrane, and some are actually anchored to cellular structures. As I said before, integral proteins are actually part of the membrane structure. They will span the entire length of the membrane and kind of poke out on either side. They can't be removed. Peripheral proteins, on the other hand, are bound to one side of the membrane surface, so they'll either be on the extracellular surface or the intracellular surface. And again, these proteins have very specific functions. They might act as receptors for different molecules or ions. They might act as channels so that things can pass through freely. So here's a really nice protein channel. Water doesn't flow freely through the plasma membrane. It might leak through. However, there are special protein channels for water so that it can pass through more readily. And those protein channels are known as aquaporins. Aqua meaning water, porin is relating to a pore or a channel. Anytime you hear a word that ends in IN, like albumin, globulin, aquaporin, hemoglobin, that usually is indicative of the fact that the molecule is a protein. We mentioned previously that on the surface of the cell externally, there are glycoproteins and glycolipids. These carbohydrate molecules help to form a glycocalyx, which literally means a sugar coating, where the carbohydrate portions extend beyond the surface of the plasma membrane, forming a sugar-coated layer, which you can see on the upper right-hand side. All these little tiny extensions are carbohydrate molecules, either bound to proteins or lipids. And this glycocalyx is important because it helps attract water, which provides lubrication and protection. The carbohydrates also help to anchor and sometimes move things. They act for specificity in binding things, and they're incredibly important in our bodies being able to recognize self versus non-self. If a person has any kind of foreign cells placed into his body by way of an organ transplant, the body's immune system actually looks at the glycocalyx on the surface of the cells to determine self versus non-self. The plasma membrane is known as semi-permeable or selectively permeable. And permeability is going to determine whether or not things can pass through. If something is impermeable, nothing will pass through. If something is permeable, everything can pass through. However, semi-permeable or selectively permeable means that some things will pass through and some things can't pass through. And this is based on a number of factors, including the size of the molecules trying to get through the membrane, whether or not these molecules carry a charge, how many molecules are on one side of the membrane versus the other side of the membrane, and that will be referred to as the concentration gradient. So if the gradient is much larger on one side and smaller on the other, things will tend to flow from high concentration to low concentration. Temperature can be a factor in permeability, and lipid solubility. Remember that fat molecules can pass through a phospholipid bilayer very readily. So if we look at substances that can move through our phospholipid bilayer, that includes nonpolar molecules because they're fat soluble, so fatty acids. Certain vitamins are fat soluble. That includes A, D, E, and K. Steroid molecules can pass through the phospholipid bilayer. So cholesterol could pass through. Steroid hormones can pass through. Oxygen, carbon dioxide. Water is polar. However, as I mentioned previously, there are special protein channels, and that helps water move through. Water can also leak through. However, aquaporins are the main reason water can get in and out of cells. Substances that can't move through without assistance include things like ions. We have different concentrations of ions inside and outside the cell membrane. We tend to have a higher concentration of sodium in the extracellular fluid and a higher concentration of potassium on the inside in the intracellular fluid. There is some sodium intracellularly and some potassium extracellularly. However, there's a concentration gradient when we look at what's on the outside versus what's on the inside. Calcium, hydrogen ions, those are all things that normally don't move through without the assistance of a protein that will either allow them to leak through or move through, and we'll look at that a lot more closely. Amino acids, polysaccharides, glucose, nucleic acids, those are things that normally can't move through. In order to move substances in and out of cells requires two different types of mechanisms referred to as passive transport and active transport. We're going to discuss passive transport first because passive transport requires no external cellular energy. Normally, atoms and molecules have their own little vibrational movement known as Brownian motion. Passive transport does not require the input of additional energy from the cell for substances to move. And as these things randomly move, they have a tendency to move from areas of high concentration to lower concentration. That's just the normal pattern of Brownian motion. If we look at this picture in the center, you'll notice that there is a high concentration of red molecules on the left and a low concentration of red molecules on the right. And via Brownian motion, with no additional cellular energy in the form of adenosine triphosphate or ATP required, these molecules are going to have a tendency to move from an area of high concentration to low concentration. They're going with or down their concentration gradient. Think of a child on a slide, okay? Kid at the top of a slide slides very easily down the slide. So going down or with the concentration gradient, just nice and easy. With active transport, that is the process of moving substances against their concentration gradient. That requires cellular energy in the form of adenosine triphosphate, and this process moves substances against their concentration gradient. So now those substances are going from an area of low concentration to an area of high concentration. Imagine that same child that went down the slide, now having to climb back up the slide. That's going to require more energy. Here we have some examples of molecules going down their concentration gradient from an area of high concentration to an area of low concentration. Those are the first two pictures on the left. High concentration to low concentration. However, the picture on the far right, we have substances being moved from low concentration to high concentration, which means we need energy in the form of adenosine triphosphate. One of the types of passive transport is diffusion. Diffusion requires no cellular energy in terms of adenosine triphosphate, and it's the result of just random motion and collisions that occur between ions and molecules, which we refer to as Brownian motion. It relies on a concentration gradient, so the molecules will spread from areas of high concentration to low concentration until they reach equilibrium. This is important because that helps to eliminate local concentration gradients, unless they are desired, in which case the cell actually has special proteins to maintain those concentration gradients. If we look at the examples of simple diffusion, here you can see a dropper with a red dye at the top and the molecules spread out over time, as well as on the lower left, you can see a tablet of purple dye has been placed in a beaker of water, and as it dissolves, the molecules tend to spread and reach equilibrium. There are different factors that can affect the rate of diffusion, including things like distance. The shorter the distance, the farther things have to travel, so diffusion can occur faster. The smaller the molecule, the faster the rate of diffusion. Imagine a little car getting into a space a lot more easily than a real large car or an SUV or a truck. The higher the temperature, the faster the rate of diffusion. The higher the size of the gradient, if there's a huge concentration gradient with a lot of things in one area versus very few in another, the larger the gradient, the faster the diffusion. Electrical forces can affect diffusion rates because opposites attract to one another and like charges repel, so that can either accelerate or reduce rates of diffusion. When we talk about diffusion across the plasma membrane, it can only be done in two ways, either by crossing the lipid portion, which fat-soluble molecules such as carbon dioxide and oxygen can do, or by utilizing a protein membrane channel. I already discussed aquaporins acting as a membrane channel, and there are other membrane channels that can allow things to pass through. So simple diffusion for nonpolar fat-soluble molecules would allow not only oxygen and carbon dioxide to go through, as we mentioned before, fat-soluble vitamins A, D, E, and K, sterile molecules, fatty acids, alcohol can go through the plasma membrane without any assistance because it can go through that lipid portion. Usually when a protein channel or protein carrier, because some proteins can actually help carry molecules across, when those types of proteins are required for getting substances from one side to the other across the plasma membrane, we refer to that as facilitated diffusion. Facilitated just means made easier. So this is diffusion made easier with the help of a protein. And different water-soluble molecules like glucose, amino acids, ions, sodium, potassium, chlorine can be moved across the membrane without the help of adenosine triphosphate, and they do this either by binding to integral protein carriers which change shape when they form a bond. When atoms join together and form a bond, it often changes the shape as we saw in proteins. So when we bind different atoms to these proteins, it may change their shape and actually help move these substances across from one side of the membrane to the other. So they can either move by binding to integral protein carriers, or they may move with water through aquaporins, and the diffusion rate will be limited by the concentration gradient and the available carriers or protein channels. When proteins are used specifically to transport substances from one side of the membrane to the other by carrying them, that is referred to as carrier-mediated facilitated diffusion, which uses integral proteins that are specifically designed for transporting polar or large molecules. The shape of the molecule being transported must fit the shape of the protein, and once the molecules or substances bind with that specific shape, the binding of that substance changes the shape of the protein, which then allows it to be released on the other side. Any kind of diffusion, including carrier-mediated facilitated diffusion, will go down a concentration gradient or with a concentration gradient, as opposed to against a concentration gradient. One example of carrier-mediated facilitated diffusion would be moving glucose into cells. It is limited by the number of carriers and would be considered saturated if all the carriers are engaged in the act of carrying molecules into the cell or out of the cell if it's a different kind of substance.
Channel-mediated facilitated diffusion is where an integral protein acts as a channel to allow substances to pass. Water goes through aquaporins, and it's not unusual for ions, which are atoms that carry a charge, either positive or negative, can get through these water channels or aquaporins, and they will diffuse down or with their concentration gradient. So we see pictures of sodium diffusing with water down its concentration gradient from the extracellular fluid to the intracellular fluid, while potassium is diffusing down its concentration gradient or with its concentration gradient from intracellular to extracellular. And channels can be selective due to the size of the pore, and they can also be selective in terms of the charges of amino acids that make up the proteins that line the channels. Leakage channels are channels that are always open. Gated channels can be controlled by chemical and electrical signals. We'll see more of that in the future. Channels can be inhibited, they can be saturated, and they are specific. Previously, we've discussed diffusion in terms of a solute, something dissolved in a liquid. Osmosis is a special type of diffusion where a solvent is moving. In the case of the human body, we're looking at water. It occurs across a membrane that is selectively permeable to water. In this particular case, we know in plasma membranes that there are different things embedded in the plasma membrane that allow not just water but solute to move. Just looking at osmosis, in this case, we've got 2 beakers of water. You see the beaker on the left has purely water. The beaker on the right has water. If there were no purple hexagons in the beaker on the right, the water levels would be equal. As soon as solute is added on the right side, as denoted by the 3 purple hexagons, water is going to move toward the solute because the solute on the right has taken up the space, and water will diffuse down its concentration gradient from an area of higher concentration of water to an area of lower concentration of water towards solute. Salt is a great example of solute. So if 3 large crystals of salt were put in the beaker on the right, water would flow across that selectively permeable membrane that only water can pass through from left to right. You'll notice that that raises the level of water on the right side. When osmosis occurs, it creates pressure known as osmotic pressure and can even go against gravity. This will continue until equilibrium, which is concentration of water being equal on both sides, is reached. And you already know that water travels through special protein channels called aquaporins. Again, osmosis is the net diffusion of water through a selectively permeable membrane from an area of high concentration of water to an area of low concentration of water. This picture actually, in my opinion, shows it a little bit better. If we have 2 areas separated by that selectively permeable membrane, you'll notice that solute takes up space on the right, and once water is allowed to flow, you see the picture on the right, how water molecules flow from left to right so that we have equal concentrations of water on both sides that is reaching equilibrium, creating osmotic pressure that will go against gravity. Water follows solute. It will follow wherever it goes. Water really likes solute. Oh yeah, it really likes solute. Water loves solute. Water follows solute. It will go against gravity. Water creates osmotic pressure. 'Cause yeah, it really likes solute. Water loves solute. Sorry, had to do that. Okay, osmosis factors. Osmolarity, if you've ever heard that term, means the concentration of solutes in a solution. Osmotic pressure is the tendency for water to move into the cell and the pressure it creates by doing so when osmosis occurs. Osmotic pressure can be opposed by hydrostatic pressure, which is the back pressure exerted by water against a membrane. Let's take a look at osmosis versus diffusion. With osmosis in the top picture, we see our semi-permeable membrane. The solute is identified as these turquoise circles, and we can see that water flows from the right to the left, going against gravity, creating osmotic pressure as the force to move water in that direction and against gravity. Diffusion, we have our solute can move now. In osmosis, the water's moving. In diffusion, the solute's moving. And so as these solute molecules can now cross through that semi-permeable membrane, we see the solute is diffusing until it reaches equilibrium, and we have a fairly equal number of solute molecules on both sides. This is an ever constantly changing state in our bodies. These are just examples. This slide is supposed to be a picture of an osmosis experiment, and if it were, the little dialysis bag containing solute, it would show that there's water going into that bag. That really doesn't look like anything's going on here except for the little concentration red dots are going out of the bag. So I'm sorry, I can't explain this picture any better. I should really probably just delete this slide. Ignore it. This is a picture that makes sense. This is a picture of a diffusion experiment. And with this picture, you can see, if you look closely, there are little green dots in the dialysis bag on the left, and they diffuse out of the semi-permeable membrane, which allows them to go out of that dialysis bag, and you can see them having reached equilibrium on the right. Why is any of this important? Tonicity is the ability of a solution to change the shape of a cell by altering the volume of water inside the cell. Remember, if the shape changes, the function changes. Structure, function. If we look at the picture to the right, at the very top you'll see a normal red blood cell in what's called an isotonic solution. Iso, that prefix means same. Tonic refers to tone. That means that the solute inside the cell is at equilibrium with the solute surrounding the cell. So there is no net movement in and out of the cell. stuff does move in and out of the cell, but there's no net movement of water, and that allows the cell to retain its normal shape and function the way it's supposed to. We know that there are different solutions that are isotonic to red blood cells, including a 0.9% sodium chloride solution and a 5% glucose solution. Those are isotonic to our red blood cells. What happens if we put a red blood cell in a solution? Notice we're using tone to describe the solution. If we put a red blood cell in a solution that is hypotonic, hypo means under or below. If the solute on the outside is less than the solute on the inside, I'm putting little dots. There's the solute outside. Here's the solute inside. If the solute is less outside than what's inside, remember, water follows solute. So the water will go from the outside of the cell into the inside of the cell, and the cell will gain water until it bursts. So it'll swell up and burst, or lyse, because the word lyse means to break or burst apart. If we put the cell in a solution that is hypertonic, that means the solution, and remember we're always talking about the solution has more solute outside because it's got hypertone, more solute. Solution has more solute than the cell. So which way does the water go? Water follows solute. The water goes out of the cell, which you can see by the green arrow, and the cell shrivels up like a raisin. It shrinks. That is called crenation. We say the cell crenates. One way that I remember what happens in what different solution is that for me, I remember hypo, hippo. If I put a cell in a hypotonic solution, the cell swells up like a big hippo and could burst. Okay, so it's potentially going to do two things, swell and burst. Active transport, as we already know, is going to require cellular chemical energy in the form of adenosine triphosphate, or ATP. Active transport also requires carrier proteins that are going to combine with the substances being transported. And you'll notice when you look at this picture, this is a classic example of the sodium-potassium pump, where 3 sodium ions are being pumped to the extracellular fluid outside the cell, while 2 potassium ions are being pumped into the cytoplasm, where we have our intracellular fluid inside the cell. This is against the concentration gradient for both of these ions. And you'll notice that energy is provided by adenosine triphosphate. Adenosine triphosphate is that molecule that has 3 high energy phosphate groups. The energy is actually in the bonds, and the bond between the second phosphate group and the third phosphate group has a lot of energy. When adenosine triphosphate is hydrolyzed and that bond is broken apart, adenosine triphosphate turns into adenosine diphosphate. The energy that's released is used to do the work of pumping these ions against their concentration gradient. Primary active transport, as we said, comes from the hydrolysis of ATP. We pump ions against their concentration gradient. I already noted that the sodium-potassium pump is going to move 3 sodium ions into the extracellular fluid where the concentration is already higher, and 2 potassiums into the internal area of the cell in the cytoplasm where the concentration of potassium is higher. So we get 2 potassium coming in, 3 sodium going out, both against their concentration gradients, and the rate of movement depends on the intracellular concentration. It is crucial to maintain a higher concentration of sodium externally and a higher concentration of potassium internally. This is important for nerve impulse conduction and muscle contraction. We'll see this in another example as we get into those body systems. Secondary active transport is a unique situation because the transport is driven indirectly by ionic gradients, and the best way to describe this would be to show you how when glucose is brought into a cell through carrier-mediated facilitated diffusion, it's not uncommon for sodium to be dragged along. It's getting a free ride. And as sodium leaks into the cells, we need to maintain sodium outside the cell. We just said that in the previous slide, and it's important for our nerve cells to be able to do this, and our muscle cells, because they have to maintain a resting membrane potential, which is the electrical difference between the ions outside the cell and the ions just inside the plasma membrane. There's a voltage that's created when we have a difference in electricity on both sides of that membrane, and it can actually be measured in millivolts. If we look at this example with glucose being transported in through carrier-mediated facilitated diffusion, when sodium hitches a ride, it has to be actively pumped out, and that's why they call this secondary active transport, because we have to transport the sodium that's leaking in as a result of the glucose and other amino acids that come in during the digestive process. Endocytosis. Endo means inside. Endocytosis means a condition of bringing something inside the cell. Endocytosis comes in three forms: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Endocytosis is a process that does require energy. So all three forms of endocytosis are going to require adenosine triphosphate in order for this to occur. Receptor-mediated endocytosis is a type of active transport where a special molecule, a signaling molecule called a ligand, binds with a receptor protein on the surface of the plasma membrane. The binding of the ligands to the protein receptors is going to cause that membrane to invaginate and form a little vesicle. I always think of vesicles like Tupperware containers. You'll notice that the vesicle has a protein coat on the outside, those little purple dots, and those proteins are going to be recycled and sent back to the plasma membrane to be reused. This uncoated vesicle is going to fuse with another vesicle called an endosome. Endo means inside. And once these two fuse, one of two things will occur. The fused product will either be transported back out of the cell. In that process, when something passes through a cell, is what we call transcytosis. Trans means through or across. Or this product may fuse with an organelle known as a lysosome. Lysosomes are organelles that contain special digestive enzymes to break things down. As we've seen so far, proteins are really important. We know that proteins have 4 levels of structure: a primary chain of amino acids, a secondary structure, which is either an alpha helix coil or a beta pleated sheet, a tertiary structure where a globular protein is formed, and a quaternary structure where two or more tertiary structures join together as a functional unit. When proteins aren't built correctly, and that's usually because the amino acid sequence is messed up somewhere, that can cause problems in the body. For instance, cystic fibrosis is a disease process that occurs when the normal proteins that are supposed to move chloride ions out of the cell don't do their job, and chloride ends up staying inside the cell. What happens is people who suffer with cystic fibrosis, which by the way is a chronic, progressive, and often fatal genetic disorder, remember DNA, that's where the instructions on how to build proteins, that's our genes, right? So this is a genetic disease of the mucus glands in the body. So if the chloride ions cannot be pumped out, what happens is a really thick, sticky mucus starts building up on the outside of the cell. Normally, if the chloride ions go out of the cell, water follows solute, so the mucus is not too thick and sticky. So this affects the respiratory, digestive systems in children and young adults, and unfortunately, people with this tend to have a lifespan of about 40 years.